Okay, welcome back to the channel. And today again, we're going to be diving into some forensic topics. So, question: um, How were crimes solved? How was forensics used to help crime solving before DNA technology? Before advanced scientific methods, the importance of observation um, was emphasized. So, keen observation was key in solving crimes. Investigators depended on their ability to notice details in the environment and the behavior of suspects. Witness statements were crucial. Detectives would interview witnesses to gather descriptions of suspects and events surrounding the crime. A skilled investigator could pick up on inconsistencies in stories or subtle cues that indicated something was off. The emphasis on careful observation laid the groundwork for building a strong case. Now there's fingerprinting. This is one of the earliest forensic methods that were available. The uniqueness of fingerprints was recognized in the late 19th century, with pioneers like Sir Francis Galton and Edward Henry developing classification systems. By the early 1900s, police departments began using fingerprints as a reliable form of identification. Investigators collected fingerprints from crime scenes and compared them with known samples, establishing connections between suspects and criminal activities. This technique became a cornerstone of forensic investigations, providing clear evidence to link individuals to crimes. Third, we have ballistics and firearms examination. Ballistics, the study of projectiles and firearms, was another critical method used to solve crimes involving guns. Early forensic experts analyzed bullets and cartridge cases to determine which weapon was used in a crime. Investigators examined the unique markings left on bullets by a gun's barrel. Each firearm has distinct features, allowing forensic experts to match bullets found at a crime scene to specific weapons. This method became essential in homicide investigations, helping law enforcement connect suspects to shootings. Fourth, we have blood typing and serology. Before DNA, serology focused on analyzing blood and bodily fluids. Forensic scientists could determine blood types using the ABO system, which categorizes blood into four main types: A, B, AB, and O. When blood was discovered at a crime scene, investigators collected samples and performed blood typing. While this method wasn't as precise as DNA analysis, it provided valuable information that helped narrow down potential suspects and link them to criminal acts. Fifth, we have hair and fiber analysis. Hair and fiber analysis was another important method in crime solving because it helped investigators collect hair samples from crime scenes and examine them under a microscope. While hair analysis couldn't definitively identify a person, it could indicate whether the hair came from a human or an animal, and link a suspect to a crime scene based on matching characteristics. Fiber analysis worked similarly, helping investigators compare textiles found at a crime scene with those owned by suspects. Next, we have document examination. Forensic document examination involved analyzing handwriting, signatures, and materials to uncover forgeries or identify authorship. Document examiners scrutinized signatures and handwriting samples for unique features. In cases of fraud or counterfeiting, this method was essential. investigators could determine whether a signature was genuine or forged, providing critical evidence that supported the case against suspects. Seventh, we have psychological profiling. Before DNA, psychological profiling was also used, although it was less common than today. Criminal psychologists analyze crime scenes and suspect behaviors to create profiles that could predict motivations and future actions. These profiles help law enforcement agencies understand the psychology behind certain crimes, especially in serial offenses. By identifying patterns and traits, investigators can narrow down their search for suspects and develop effective strategies for apprehension. Next, eighth, we have community involvement. Finally, community involvement was crucial in solving crimes. Law enforcement relied heavily on tips from the public, and community watch programs often helped identify suspicious activities. Engaging the community built trust and encouraged people to report crimes or unusual behavior. Investigators followed up on leads provided by citizens, which often led to breakthroughs in cases that might have otherwise gone cold. In conclusion, while DNA technology has transformed the field of forensic science, early investigators used a variety of clever methods to solve crimes. From fingerprinting and ballistics to community involvement, these techniques were essential in delivering justice. How much did DNA technology statistically increase the chances of catching offenders? And what was the general impact DNA had on law enforcement and crime and conviction rate? DNA technology emerged in the 1980s and quickly became a revolutionary tool for law enforcement. For the first time, investigators had a reliable way to match biological evidence found at crime scenes with potential suspects. Before DNA, solving cases often relied on less precise methods, like we were talking about above, leading to wrongful convictions or missed opportunities to catch the right suspect. With DNA, law enforcement could accurately identify individuals based on their unique genetic makeup, which is present in bodily fluids, hair, skin, and other biological materials. The introduction of DNA technology has had a profound statistical impact on conviction rates. Research shows that DNA evidence has contributed to solving thousands of cases that previously went unsolved. In some jurisdictions, studies indicate that the use of DNA evidence can increase the likelihood of securing a conviction by up to 50% or more. This remarkable improvement is largely due to the accuracy of DNA matching, which minimizes the chances of misidentifying suspects. Organizations like the Innocence Project have used DNA testing to exonerate wrongfully convicted individuals. Since its inception, they have helped free over 375 people in the U.S. who were wrongfully imprisoned, highlighting DNA's crucial role in correcting past injustices. Another significant advancement has been the creation of DNA databases such as CODIS, Combined DNA Index System, in the United States. These databases store DNA profiles from crime scenes and convicted offenders, making it easier for law enforcement to identify suspects. When a new DNA sample is collected, it can be compared against existing profiles in the database. This process has led to numerous cold cases being solved, where crimes committed years ago are linked to current suspects through DNA evidence. The ability to connect past crimes with current offenders has had a profound impact on crime resolution rates. The introduction of DNA technology has also created a deterrent effect. Knowing that DNA evidence can link them to crimes, potential offenders may think twice before committing certain offenses. This deterrent effect contributes to overall crime reduction, as the fear of being caught through DNA evidence can influence criminal behavior. Studies have shown a decline in certain types of violent crimes, suggesting that the presence of DNA technology has had a broader impact on crime rates. Now, there are some challenges and ethical considerations of DNA. While DNA technology has greatly advanced law enforcement, it also raises important ethical considerations. Issues like privacy, consent, and the potential for misuse of genetic information are critical discussions in today's society. Additionally, as technology advances, the possibility of identifying suspects based on distant familial connections through techniques like familial DNA searching creates new ethical dilemmas. Balancing the benefits of DNA technology with individual rights remains a vital conversation within the criminal justice system. Conclusion: DNA technology has dramatically transformed law enforcement, increasing the chances of catching offenders and securing convictions. Its accuracy, the establishment of DNA databases, and its deterrent effect have all contributed to its success in solving crimes and delivering justice. Now, I'm curious what some of the flaws and shortcomings of DNA evidence in forensics and law enforcement are, and times it was used incorrectly, leading to wrong arrests, convictions, or jail times. It's important to understand that DNA evidence is not infallible. Although DNA is unique to each individual, the process of collecting and analyzing it can introduce errors. One major issue is contamination. DNA samples can be easily compromised at the crime scene, in the laboratory, or even during transport. If a sample is contaminated with DNA from another source, it can lead to inaccurate conclusions. For example, if a technician accidentally mixes samples or collects evidence without proper precautions, the results can be misleading. Another significant challenge is the interpretation of the DNA evidence. DNA testing results can vary based on the method used and the skill of the forensic analyst. Statistical calculations often accompany DNA results, estimating the likelihood that a random individual could have the same DNA profile. However, these probabilities can be misinterpreted or miscommunicated. If a jury hears that there is a 1 in a billion chance of a match, they may mistakenly assume that this guarantees guilt, overlooking the nuances involved in the analysis. Familial DNA searching is another complex area. This technique involves analyzing DNA to identify potential relatives of a suspect. While this can be helpful in solving crimes, it raises ethical concerns and can lead to wrongful accusations. For example, a person might be implicated in a crime solely based on a familial match, even if they had no direct involvement. This practice can lead to innocent individuals being drawn into investigations without sufficient evidence against them. Next, we have human error. Human error is also a factor in the use of DNA evidence. From sample collection to analysis and reporting, multiple opportunities exist for mistakes. Forensic scientists are human and can make errors in their work, whether due to fatigue, miscalculation, or oversight. Mislabeling samples, misinterpreting data, or failing to follow proper protocols can all lead to incorrect conclusions. These human errors highlight the importance of rigorous training and quality control in forensic labs. Thanks for watching as we dived into DNA and its use in forensics, and we'll dive more into it in the next video.